Yeah, so my name is Laurent. As Steve said, I'm a postdoc at the MBL. Today I'm going to present the topic of my postdoc, as Steve said, about recording data provenance from sample to workflow result with LabID. Um, Before I start, a quick word about the EMBL, which stands for European Molecular Biology Laboratory. It's quite well established life science research institute with 6 sites across Europe in 5 different countries. Research at the EMBL covers a wide diversity of modalities and instruments, I would say. So we have X-ray, light and electron microscopes, but also sequencer for genomics projects. So quite some wet lab research, but also quite strong bioinformatics expertise and computational infrastructure. So there's a Slum cluster available. As part of the Data Science Center, we do trainings for researcher, but also consulting for data management, data analysis, and also development and maintenance of services, such as Jupyter Hub, Galaxy, GitLab, or Lab Integrated Data, short LabID, which is our data management platform, and which will be the focus of this presentation. By Galaxy here, I'm obviously referring to the workflow management or workflow engine. So we also, which is quite popular in our community for bioinformatics, and the MBL is also running its own instance of it. Now short about LabID. So this is a all-in-one fair data management platform for life science institute and research group. It's a kind of central platform for researchers to drop their data together with metadata and kind of interconnect it and share it within their research group or with colleagues. This is an open source solution that's been developed at the EMBL Heidelberg over several years now, and it's used across different EMBL sites and now kind of trying to expand to other institutes. It's very much tailored to life science, but there are many kind of generic data principles, so I hope there are a few bits that would be of interest for you. So the idea of LabID is to capture metadata at several steps of the data life cycle. So if we look at the traditional kind of biology experiment, this will start with the biological specimen from which some sample would be extracted, and the sample would be then analyzed via some assays. So might be some microscopy imaging, for instance. As output of this assay would be some raw data or primary data, and eventually later on this primary data would be processed by some workflow. So this is kind of the data flow we have in biology. So as part of the assay, there will be also some consumables and protocol involved. And basically, these are all kind of entities that we reflect in our system. That's how we kind of modeled the problematics here. So the platform is quite valuable. Oh yeah, then there's also an electronic lab notebook available for researcher who can kind of directly mention all of these entities in their notes, and yeah, keep it quite also user-friendly in a way. The platform has kind of several benefits and applications. One of them is facilitating the position of data into repositories, so research data repositories. In our communities, it would be, for instance, ENA for genomics data or biostudies, or the bioimage archive for microscopy images. And all of these kind of repository also expect quite some level of metadata. So from kind of any steps of this work stream, let's say. And so the LabID platform makes it easy to kind of compile this information for submission. The platform also makes it easy for researcher to work with collaborators, but also within their own research group, basically to keep track of the data that was generated and the context around it, and to kind of nicely organize it, and to have kind of any level of metadata they need to be able to communicate and collaborate on projects. It's also quite valuable to kind of annotate and consolidate data set for machine learning or AI application, especially since the platform can also be leveraged with the machine actionable. Yeah, we have some kind of command line interface basically that allows to do a bit of automation around in this direction. So there are kind of 4 interconnected modules in LabID, which, let's say, target maybe different user segments. So and in the interface, they are also kind of ordered in the way they would appear in a biological experiment. So starting with kind of lab collection management, so consumables, equipment. These are more for like lab manager would usually deal with this section of the platform, while researcher would be more dealing with biomaterial management. Let's say if when they run an experiment, they will use a specific cell line, maybe also specific antibodies. So those would be also reflected in the platform. They also will make use of the electronic lab notebook so they can describe their experiments and protocols. And then the outcome of this would be some data sets, potentially some workflows. So all of these kind of are nicely described in the system and can be interconnected. And it's kind of also unified user experience around those different but related thematics. So if I take now an example of such a biological assay, let's say, might be some tissue imaging. So as I say, we will start with a specimen, maybe a specific transgenic line of a mouse, which has like really a reference, maybe even published somewhere. From this sample, we would extract some biomaterial, maybe a tissue section. And this tissue section, we could image it on a microscope, for instance. Let's do some histology imaging, for instance. And as outcome of this imaging would be some raw data. In this case here would be some images. So again, each of these kind of entity would be reflected in the system and can be interconnected. And once this is done, we have like a nice visualization of kind of the data provenance all the way to the kind of original specimen. So we would have one entity for the transgenic line. We can see that from this kind of mouse, we extracted 3 different samples, which were all imaged as part of the same microscopy assay. And as a result of this assay were a couple of images, maybe some metadata also about the acquisition, or maybe even some reporting. So this is kind of nice end-to-end from sample, like biological sample to data. So the way we also ensure that we keep the biological provenance when registering data in the system is that we have kind of a wizard when researcher basically drop the data in the system, kind of a step-by-step wizard for which they have to basically associate this data to an assay. In this case, this would be our microscopy imaging. So this assay would have a reference to an instrument, also referenced in the system. And as input to this assay would be a sample. So as I said before, maybe like a tissue section or so. The raw data would then be part of a study, which would basically a collection of similar data set, and this study might be part of a bigger project. So let's say your postdoc research project, for instance, or PhD project. So this kind of a way to model the biological provenance is very similar to the ISA framework, if you have heard of this, which stands for Investigation Study Assay, which is kind of a proposed framework to describe biomedical research data and provenance. All right, so the story does not stop here. Once we have this raw data, typically it's processed using a workflow. Let's say Galaxy, for instance. Another result of this, we get additional data, so derived data. So before this project, the kind of state of the art of our platform was that we were able to document the provenance of this data by just connecting it to the raw data. So then we had kind of this biological provenance in place, but we were kind of losing all of this computational provenance. So we're not saving any information about the workflow, like what version was executed, what were the parameters used. So we're losing quite a bit of reproducibility, and yeah, we're not really fair compliant in a way. So this was one of the motivations for the workflow integration of LabID. Which had kind of with subtitle documenting the provenance of derived data. This project got funding by the European Union as part of the OSCAR Research Consortium with the official title LabID Prov for provenance, tracking and sharing data provenance with EuroCrate in lab integrated data. All right, so a bit more detail about motivation and goals for these projects. So as I said, in our communities, workflow are commonly used to process data. So as I said before, Galaxy, but also increasingly Nextflow and SnakeMake. But despite the kind of broad usage of those workflow, increasing use, there was not like a one central solution to track workflow execution. So kind of each workflow engine has its own way to do things. So in Galaxy, we'd have a nice dashboard where you see what workflow were executed. Nextflow would provide some nice HTML reports, for instance. But there is no like central solution to have an overview of what workflow were executed and what was the data associated. Besides that, we also realized that workflows are not systematically published. Neither is the derived data, so the data coming out of those workflow, or it's kind of community dependent or application dependent. And when this data is actually shared, it's often lacking kind of critical metadata around the research context or about the biological provenance. So let's say if we look at maybe a processed image, do we know from which sample it was derived? So the question was, how can we simplify tracking and sharing of derived data with kind of full data provenance, so both biological and computational? And our proposal was to use LabID to document workflow execution, kind of to build upon the capacities of LabID to document provenance, to extend it to workflows. So basically extending the current graph, knowledge graph that we have in the platform to this derived data. Then to integrate with workflow repositories such as Workflow Hub and workflow engines, let's say Galaxy, Nextflow, and SnakeMake, as these are the most used in our community. And finally, leverage the AeroCrate specification as an interoperable format to capture both biological and computational data provenance. So AeroCrate stands for Research Object Crate. I will come back to it in a moment. The expected benefits of the project are to foster fact practices around workflow, also to facilitate collaboration between user and developers of workflow, and also to encourage, as I said, publication of workflows and workflow runs. Okay. Starting point for the project was the question of how we want to model like workflows and workflow runs. And so the way we did it is we have a workflow, which is basically kind of a top level entities for which we can have different version. So we kind of distinguish workflow from a version. And when we talk about workflow runs, we are really talking about one specific version that was executed with some input data, specific parameters in execution environment, and which generated some output data. So all of this would be the workflow run. So this is how we kind of model it in our platform. Now coming to the workflow versions. So how can we create or import workflows in LabID? So we implemented different modalities to do that. The first one for Galaxy, we used the Galaxy API, the so-called BioBlend API to directly be able to import workflows. So it's quite straightforward. Then the way it looks like in the user interface is that user can directly copy paste the URL to a public workflow, or they can from the LabID interface have like a small search query and directly import from there. So this is quite straightforward, and it's kind of an illustration of how we can leverage an API from a workflow engine to integrate with LabID. The second way to get workflows into LabID is to import from Workflow Hub, for instance, and there we are leveraging the Workflow AeroCrate, which is a kind of generic format to share workflows. And so in theory, we would be able to import from pretty much any repository which can provide a generic workflow AeroCrate. And there, again, the user experience is quite straightforward. They can select from which repository they want to import, and again, either copy-paste a URL to a public workflow or do a direct search of these registries and import it within LabID. So this is quite nice. So one before last option is to import using the Git integration. So if you have your workflow already in GitHub or GitLab, can be a private repository, or let's say institutional. So we have some support for authentication. Then it's also very straightforward. You can copy paste the URL to your Git repository, select the version or a specific commit, and then LabID will kind of list or kind of discover automatically which file seems interesting, and you can kind of refine this selection and select which file you want to import. And here again, we keep track of what version was imported, so the URL and the commit will be stored in our system, again, to keep track of the provenance. The last option for researchers to kind of put together a workflow in LabID is just by basically drag and dropping files from their machines. So this is quite user-friendly, and for researchers who are not familiar with Git or for which workflow is not publicly available, this is a quite nice alternative. So as I said, they just have to drag and drop and select like a type kind of to indicate if it's maybe like entry point file of the workflow, for instance, or more like a configuration file. Under the hood, we are actually also using a Git repository, a local Git repository, to just be able to, so we use this kind of internal Git repository as a way to do versioning, so that user can very easily kind of freeze or release such workflow version that they made from local file and make a new version. So they don't even realize this is a Git repositories, but they can do kind of simple workflow versioning via LabID. Okay, regardless of how the workflow was imported or created in LabID, we also support exporting workflows from LabID. And here again, we are leveraging the workflow error crate specification as a standard, and we support direct export to Workflow Hub. Or you can just get basically a zip of this workflow error crate and publish it or make use of it in any other platform that would be able to take advantage of this specification. Quick word about this publication to Workflow Hub. It's very straightforward again here. It's kind of just a one click in the interface. The user just need to kind of configure their Workflow Hub API key in LabID, and then it's stored, and then they can very easily publish to Workflow Hub or to the dev Workflow Hub for testing, basically. And again, we store a reference to the published workflow, so that if it's reused as part of some workflow, and we also know that this was a published workflow. Okay, so far this is a bit the picture. I've shown how we can import and export workflow version in LabID using different modalities, so either with an API or leveraging the workflow Hub specification. Now, what about workflow runs? So here we have kind of a similar user experience that we can, if you run some workflow in Galaxy, we also leverage their API to directly import Galaxy invocation into our system, which then makes it easy again to connect it to the rest of the provenance available in LabID. For other workflow engine, if there is the possibility that the engine generates a so-called workflow run error crate, which is another kind of declination of error crate for workflow run, so basically a package of the workflow and all the data involved in the run. We implemented in our command line interface kind of a parser for such package. And from this package, then we are able to extract automatically basically the list of files or kind of data set that were used as input, used as output, eventually even extract additional information such as what version of the workflow was used, what was the execution environment. So this is a kind of a very straightforward, there is little configuration needed, basically. If the workflow engine does not, so currently Nextflow can generate such kind of package using the NFProv plugin. Galaxy is also able to generate such kind of crate. I think there is work in progress also for SnakeMake. So yeah, it's then quite straightforward for us to kind of ingest this kind of data. If such object is not possible, we also have the option to kind of automate data registration for this workflow via, again, our command line interface. And the way we do this is by what we call so-called sniffer, which are kind of customizable, pluggable Python classes, basically, which are configured to basically look for specific files, maybe like looking for specific extensions and registering those as input and other extension as output to the workflow run. And basically, yeah, leveraging the REST API of LabID to do the registration. The last option to register a workflow run is to basically do it via the user interface, and there it's also quite user-friendly. So they just have to register the data via the wizard I showed before, and once the data is in LabID, they can quite easily link this to a workflow run. So it might be more for user who are maybe not so much versed into computation or playing with the command line executable. So let's look at an example of workflow run in LabID. So we're looking here at an image conversion workflow. So the input was some images like in a proprietary format, so like some CZI image from a Zeiss microscope. So this is this entity here. And this kind of proprietary format was converted into an open format, so OMISR, which is quite popular for microscopy. And so here we have both the provenance of the biological provenance, so all the way to the sample and specimen. And so that we can see that those two MEs are kind of derived from this kind of top level data set. And on the other hand, we also have the computational provenance on the left. So we know like what version of the workflow was executed, eventually additional metadata about the execution and which data set were generated as input and output. If this workflow was registered via the command line interface, we could also think of automating the registration of such workflow run after conversion, like just adding an additional kind of job after conversion to automate this task. Another workflow run, this is here a Nextflow workflow. So this is again how the workflow looks like. So as I said, we always have a reference to the workflow version that was executed, even eventually the status of the run. So the platform might even be used to kind of even document failed workflow. So if you can integrate with external system, then you could use the platform again to kind of do this tracking. And then for a given workflow run, this is a bit specific to LabID, but the data associated to the run can be assigned to kind of different types. So the simplest one are the inputs and the outputs, but we can also have more specific types like reports and log, which you could say are more specific output types, and for which we can maybe leverage a bit nicer representation. So if we look at the output dataset tab at the bottom, this is basically the list of dataset that were generated as part of this workflow run. Yeah, so it's quite nice visualization of a workflow run. Okay, so now we are quite happy about the situation. We are able to track the provenance, both biological provenance and computational provenance in our system. The question now is how can we share or export this kind of graph of data provenance for publication or for, let's say, interoperability with other system. So until now, we had support for export of tabular data. So it's a CSV, for instance, or more specific formats when depositing to a specific research repository like ENA. But those were either quite limited in what they can express, especially for derived data. And apart from this kind of repository, there was not a widely adopted standard for tables. So we sort of then building upon the workflow run AeroCrate specification, which, as I said, is used to package data of a workflow run. And it turned out the specification also allows to describe a graph of data in so-called context entities. And by this, they mean actually metadata, which is basically also what we're doing with LabID. This specification is standardized, so it's using ontologies and control vocabularies, so which enables then interoperability and machine actionable tasks like indexing and searching those kind of research objects. And it's also extensible. So the workflow run AeroCrate itself is an extension of the kind of base AeroCrate profile. for workflow run, but there are also like other vocabularies or other specification available that can be kind of plugged in into this JSON to document biological provenance, let's say with assay and lab protocol to describe sample preparation. Another kind of attracting aspect was that the specification was already adopted by other platform and communities, such as Workflow Hub, Nextflow with the NFProf plugin, as I said, Galaxy, which was also able to generate such great, or the ARC community, which stands for Annotated Research Context. So, um, so what's happening now when we want to export workflow run from LabID? So this is kind of the big picture that I was showing before, now including the workflow run. And so there's quite some information, quite some relationship between all these different entities. But with the workflow run error crate or kind of augmented workflow run error crate, we are pretty confident that we can reflect most of the metadata available in LabID. And then basically allow people then to share and publish this information with all the provenance. So again, for the export of such workflow run error crate from LabID, it's quite straightforward. It's kind of quite straightforward, kind of one-click task, and so it's quite agnostic to the workflow engine. So any workflow run registered in LabID is able to generate a workflow run error crate for this, and also keeping kind of the reference to the LabID entities that were exported, so we can look at the error crate and back into LabID find out which entities were exported, and this idea of exporting the workflow run is also kind of transferable to other disciplines here who are using like potentially biology-oriented ontology. But again, this is a very extensible specification, this arrow crate, so the idea could be transferable to other disciplines. So with this, I'm slowly coming to an end. So I hope I could show you how LabID integrates well with the workflow ecosystem, how we can import workflow version and export them, either leveraging APIs or standard specification. Same for workflow runs. And again, the takeaways for this project was that we are trying to enhance fair practices around workflow by documenting both biological and computational provenance of data. We integrated with workflow management system and repositories. We streamlined the publication and sharing of workflows and workflow runs. We also facilitated versioning of workflows and collaborative development, and provided simple automation mechanism to kind of foster adoption of the tool. If you are interested, you are very welcome to try it. We have a demo server. Currently, it's offline, unfortunately, because we had to resolve some vulnerabilities, but it should be on again, I hope, in 1 or 2 weeks. You can also try to run it on your machine with Docker Compose. Yeah, you can have a look at the instruction in the repository. We have also quite some documentation, videos, and tutorials on our documentation websites. And we will also run an in-person training on this topic of the workflow integration here at the MBL end of September of this year, most likely. And this will be also open to external participants. As I said before, the project is open source. So we're also welcoming contribution and collaboration. So there are different ways you can contribute. So either just providing us feedback on kind of user experience, but also on admin experience. We're trying to install it on their institute. You can also contribute to the documentation, provide some suggestion or reporting some issues. The source code is available on GitLab, and you can also reach out to us either by email or via Slack, so you can scan this code basically. With this, just take a minute to present the team. So as I said, the LabID has been developed over the years at the EMBL Heidelberg by the Modis team, led by Charles Girardot. We are currently 5 at the MBL. And on this project, we also had a contribution from Yellow, who is now, so he's a former team member. He was also in the Modis team for several years, contributed a lot to the original project. And as part of this workflow integration, he also was contributed, so was contracted as part of the Oscars project. As I said, the project was funded by the EU via the Oscars consortium, and we're also very grateful to the MBL for helping us maintaining or giving us the possibility to maintain this software. All right, with this, I'm finished and I'm happy to take any question. Thank you.